Hi, John. It's good to see you.
It is good seeing you, Sean. How are you?
I am well, thank you. What is on your mind this week?
Uh, so I have a good friend that's battling cancer right now, so that's been on my mind quite a bit. He has a rare form of cancer and is in a research hospital here in the U.S. And it turns out that the type of cancer he has is what this research hospital is in the middle of researching. It was happened to be right, close to where he lived. That's where he was, an option for him to go anyway. And they are doing
Mm,
research on this really rare type of cancer. And so it's an aggressive form, but he is in really good hands, I think, because of being part of some research into trying to figure out how to combat and defeat cancer at some point. And so he's been on my mind
mm,
a lot just because of his struggles he's going through now with chemo and the different treatments and all. Just staying in touch with him and hearing what he's
right.
going through, it's really tough. So my thoughts and prayers are obviously with him. But yeah, that's what's been on my mind.
I understandable. It's interesting that you bring that up though, because well, in past episodes of this podcast, we've touched on research, and I know how important fidelity is to you. You know, when you talk about research and I think we've discussed too some flawed, do we call it flawed research, but um, and how um, maybe not scary, but how bad that can be for a researcher if they. But I feel like I don't have my head wrapped fully around research, how it's
Mm-hmm.
conducted, what decisions are made, and and so I know you've done a lot of research, so. Can you break this down? Can you explain this to me like I'm Sean? Um, at the very beginning, you know, how do you? So let's go back to the hospital. They're researching a very particular type of cancer.
Mm-hmm.
Seems to me that makes sense if they're wherever they're getting their information, that type of cancer may be on the rise or whatever jumps out at a researcher. But you have a theory, correct,
Mm-hmm.
that you want to put to the test. So how I'll ask you, how did you come up with a topic that you wanted to do research on?
Yeah, that and you know, you're saying they're researching this rare form, and something must have jumped out to them. A lot of times, researchers choose their research topic based on, uh, for some, usually a lot of times personal reasons, like maybe they have known somebody with this type of cancer and realized it's rare and there's not much research there, or there can be story. There's usually a backstory behind research, not always, but usually there is. Um, and so, yeah, my research topics were always around something that interested me or a question that I had that I was wanting an answer for whatever reason that may be. But
Mm-hmm.
it's really important. Research is really important so that, we've talked about it, kind of touched on it multiple times, like you mentioned. And now it's when it touches you like a friend that has cancer, which this friend as well. So you know him.
I
And
do know him. Yeah.
so when you're when research kind of becomes personal like that, when it's somebody and you're wanting a cure to help them out, it's like, why can't we do more research? Why couldn't we have already been doing research? Why haven't we found this out by now? And it's because it's a process that takes time, and it takes funding and time. And that's why I'm passionate about science and funding sciences. And there's real world consequences when we don't. So I think that's kind of why it's on my mind. And I think that it's also important to know what sometimes throws people off is when there's research with like mice or rats or monkeys. It's like, why are we studying those? Why can't we just help people? It's like we should be working with people. So that's always kind of an interesting point too.
Yeah, I was going to try to do this a little bit more chronologically. So, well, I'll stick with that because I want to get back to rats and mice and all of that. And I know that that's not your specialty, is, rats and rat studies, but I do want to come back to that. But again, to make this easier for me, so whatever happens, the reason is you pick a topic,
Mm-hmm.
you pick something that you want to research, and well, not always. I was going to say, and um, you come up with a hypothesis, but that's not always true, right? That you're testing.
Yeah, there's some form of testing. You're trying to answer a question. So sometimes you have a
Okay, that's a better
hypothesis,
way of putting it. Yeah,
and the
yeah.
hypothesis will be based on what you already know about it. So a hypothesis is kind of like your best educated guess at this point. And so sometimes you
Hmm.
go into something that's like, I don't know much about this. I just have some questions around it. So there can be like exploratory research to just answer some basic questions. But then eventually, if you're going to do experimental research, you have to have, some type of hypothesis that you're testing.
So there is different reasons to conduct research. As I was asking the question, it occurred to me that we've done research. I know of some of your research where you don't have a hypothesis necessarily. You're just it's an experiment to see what a group of people will do in certain conditions. Is
Yeah.
that accurate?
Yeah, there's still a hypothesis. So all experimental research has a
Oh,
hypothesis because
I was on the right track. Okay.
you're testing against the null, which the null hypothesis means that whatever you're testing is not going to have an effect. So like experiments that you've been involved in is it's where we're usually testing the effectiveness of a training methodology like preflex to see if preflex is effective or not.
Hmm.
So the null hypothesis would say you have these two groups. You have officers who have been using preflex and those that have not been using it. And the null
No.
hypothesis, there's not going to be a difference in their behaviors or thoughts or anything based, because that's the null. And so the hypothesis is that
Hmm.
there will be a difference based on and then the hypothesis usually tells the direction of the difference. Like they'll have better memories or increased recall from this, or their behavior will be more like this. And those are educated guesses based on what you already know about the topic and kind of what you expect. But you're always testing the null hypothesis. You're testing to see if there's no difference or not.
Right. And then going back to the cancer research that you mentioned in the beginning. Um, I mean, they know that this type of cancer exists. So are they actually looking at treatment,
Yes.
or what would that kind of research be designed for?
So it would be, cancer generally, attacks the body's cells and blood cells and things like that. And so, but in different types of cancers in different parts of the body will have different effects. So one treatment for one type of cancer may not work for a different type of cancer for a variety of reasons. So then you say, okay,
Hmm.
this is a rare type of cancer and it's located in this area of the body, and here's what we know about this area of the body. And so now we think that the best way to attack this type of cancer is with, method A. And so that will be what they start testing. And then usually it
Hmm.
almost always starts with animal studies, non-human animals, and then it gets approved to start trying things with humans. If there,
Human trials. I
yeah,
mean, yeah,
if there's
yeah.
pharmaceuticals involved, it's almost always started with animal trials before it gets to human trials.
And usually rats. Is that fair?
Yeah, I think in pharmaceutical studies, it's usually mice and rats, the rodents. But in general, just research in general, there's four kind of mammals that are the most selected of all, and that's mice, rats, rhesus monkeys, and macaque monkeys, I believe, are the 4.
Okay.
And there's other animals like chimpanzee studies, and there's other studies as well, but those seem to be the 4 most common animals researched. And there's reasons for that, but those are usually the 4.
Well, so that's what I wanted to ask is, it doesn't seem to me that we have that much in common from a biological standpoint with rats.
Yeah.
Like as far as our brain, our all of our circulatory system, all of that is different.
How is
that
how is that different?
Well, I mean, hey, who's the doctor here? Who's the scientist here? I feel like
Yes.
that our bodies are different enough, but that's what I should be asking maybe is, are we that similar?
So here's a way to look at it. We all live together on the planet that we're on. And I'm talking about humans and non-human animals. I'm talking about mammals specifically. So humans and non-human mammals. So we all live on the same planet together. So we're sharing things like we share air, we share water, the things that we eat, grasses, berries, fruits, meat. We share those types of things. So if we're all in a system, that means that our bodies are going to be built to, need the sun, need water, need air, need food of different types. So now when we're ingesting like food, for example, we have to have ways to break the food down and how to convert that into energy to use. So we're all, it's like having, an old Ford pickup truck and a brand new Lamborghini and a 1978 Pacer and 2002 Jeep Cherokee. All of these are vehicles. They all are built a little bit differently, but they all use fuel, like gasoline, a little bit different types of gasoline, but they're also all cars run on basically the same thing, have similar wiring. And I know it's gone from carburetor to fuel injection, but the idea is the same. It's just the system's changed a little bit, but that's kind of maybe similar to how mammals are. We're all running on really similar systems. So because of that, our brains are fairly similar, but not all of our brain though. So there's differences between mammals, but there are some key structures and systems that are remarkably similar in all mammals, and that would be like the limbic system. So all mammals have basic limbic systems. They look a little bit differently, but that's for processing emotions. They all have reward circuits for learning and forming habits and things like that. We usually talk about dopamine pathways. So they all of us mammals have these reward circuits, and we all have, a frontal cortex with a prefrontal cortex where there's decision making and inhibition of behaviors and actions and things like that. Those are really similar structures in all mammals. And then, go ahead.
I'm sorry. You sold me. We can keep going. You sold me on the medical research side of this using rats, mammals, or would you say non-human mammals? Mm-hmm. Okay, so what you say makes complete sense to me about the medical side, because we do have, I just learned, we have a lot of similarities in our systems. But what about using specifically rats, but we can talk about non-human mammals, but for behavior
we'll stick
research.
To rats. Yeah, let's stick to rats.
Okay, so
or mice. Like
so
rodents.
Using rats are fine. And just because I think that it probably resonates when you think about rats. Like, let me ask you this. Let's start with this. Is there a difference between rats that that I find out in a field and ones you find in the lab?
Yes.
Lab rats created for, there are lab rats that are created specifically for research. Is that accurate?
That is accurate, and just so that it's really uh, for research control, to tightly control experiments, so you know what their genetics are. So that's really the
Mm-hmm.
difference. Like you could take a rat that you find in a dumpster and put it in experimental stuff, but you don't know why. There might be, whatever you're testing, there might be some other reasons why a rat is responding that way based on its past learned experiences. So if you want it to be a really tightly controlled study, then you want to raise a rat in a lab rat. And there's different
Right.
kinds. There's like the Wistar. I'm trying to think. This is from memory, because I've also conducted research with rats. So there's a certain
Oh, okay.
yeah, there's certain types of
Okay.
rats, and then uh, they have fairly short life cycles, and you can order rats and mice with certain genetic mutations, so you can order them
Okay.
with certain physical conditions or even certain disorders and things like that. So yeah, they're so that's the behavior side. And then when I'm talking about the limbic system and reward circuits and those types of things, that lets you look at some of the fundamental brain processes around learning, fear, anxiety, memory, addictions, social behaviors. So that can cover medicine or just looking at behavioral changes.
Okay. So here's the question in my head, and this might be way too far out for this, but you've talked about before rat uh, this research where like the rat goes in a maze, and if they push a button, food comes out. If they push it every 30 seconds, or if they push it 100 times, or what, you know what I mean. But if you were to take me, if we could do this, throw ethics out for just a second. But if you take me and put me into a giant maze with the same structure, a human being,
Mm-hmm.
would they do the same thing or a similar thing?
Yeah, I mean, we have examples of that in real life. I'm trying to think. I'm trying to think of some examples, but like when you're waiting for a new post on, you know that you're certain people post at certain times, so you don't sit there and look. You wait
Mm-hmm.
till you know they're going to post, then you check it. That's like, I know that there's a time frame before I check my phone again, or I don't know. There's things like you know TV shows and commercials, and there's a lot of behaviors that we have that have are interrupted with timing sequences, and so it's just simple to look at like a button press and a piece of food with a rat, because you can control various things around that to see what the scheduling looks like. But then when you apply it to human behavior, you find the same thing.
Dang it. Okay, you got me on rats. Okay, this makes complete sense now. So now I want to go back because I did kind of take this out of order because I really wanted to learn about why we use rats
Okay.
to study human behavior, but I got that now. So let's go back. The chronological kind of steps in general for conducting research. I come up with a hypothesis.
Mm-hmm.
I want to test that hypothesis. My best, an educated guess, and I want to test that.
Mm-hmm.
And then what was that as far as we got? Was there a second step?
No,
So then what's next?
you,
Yeah, what's
you'd
next?
ask why you might do research, and then you have a hypothesis. Then you have to design the study to find the answer you want. And in experiments, you make sure you have two separate groups. So you have the experimental group and the control group, and it might be multiple groups. You might have a control group and 3 or 4 experimental groups, depending on what that study looks like. And then how you design it. You can design. There can be well designed studies, and then studies that are not very well designed. So you have to really, think about that and put a lot of thought into how you design it.
and not the comprehensive list, but maybe can you give me kind of the more not most popular, but like what are some? You've talked about surveys. You've talked about. I'm trying to remember all the different research you've mentioned, but what are my choices when I want to design a study?
So surveys are different because that's a little bit different. I'm talking so. With experimental, where you have a control group and a experimental group or multiple experimental groups, some of the things to look out for would be randomizing, getting a random sample of participants. We talked about what happens when you don't, when we talked about the executive monkey several episodes ago, where these researchers, they had a group of monkeys and they were going to put them into a group of executives and then those of like blue collar workers. And so they were teaching them to press a button to avoid a shock. And those that learned how to press the button the fastest were automatically put in as executives just to save time. And those that didn't learn it quite as quickly were in the blue collar group. And then they found that the executives developed ulcers and other anxiety type behaviors around for their job of having to press a button, because their button press would help avoid the shock for them and the other monkey, the other blue collar monkey. So there are two monkeys harnessed to the same equipment, where the executive monkey pushing the button would help both of them. If the executive monkey didn't push the button, they would both get shocked. And so when they developed ulcers and things, they said, oh, that's a life of an executive, which it turns out when they randomly selected them, it had something more to do with type A personality than it did being an executive. So when they randomly selected them, that effect went away. So randomization is really important. There can be other confounding variables that you don't control for, like if you're testing, like especially like in rat studies, if you have one group that you test in the morning, the other you test in the afternoon, just that time difference can lead to significant changes in behavior. So now you're really measuring differences in time rather than your treatment effect. So you have to make sure that if you do have to have a morning and an afternoon time, that you test half the control and half the experimental group in the morning and half the control, half the experimental group in the afternoon. Sample size can sometimes cause a problem. So you think, oh, I just need 10 people for this study, when really you need 50 people. Because, everybody is different, and not just people, but rats too are different. So if you are testing something, you have 10 people, maybe there's that one person that's an outlier that they're going to skew the data towards them. But if you average them into 50 people, then that one outlier isn't going to have as large of an effect. So there's statistical models that researchers use that will tell you how many people you need to obtain a certain power value for that study. So power values need to be at a certain level. And so it tells you what there's certain things you have to know and do, but you put those into a formula and it comes out and says you need 48.2 participants per group. So then you say, okay, so more than 48 you don't split people up. You would say, okay, that's 49 or 50. I'll just do 50 per group. So there's a lot of things that go into the design of the study, just to make sure that whatever results you get back, remember, as I've said, you don't want to be embarrassed by it, because other researchers are going to, if you find something, other researchers are going to also test it to make sure they can find it too. And you don't want to be kind of hung out to dry because you didn't follow the right procedures and you look stupid because of it.
Well, so again, jumping out of order a little bit, but you conduct research, you have a finding, whatever that finding might be, and then the rest of us that are in a field or a similar field are going to try to replicate that study and come up with. But so when I am doing that research, my hypothesis is that you're wrong.
Well, so
I don't want to say it in such a negative way, but that I am setting out to try to disprove your finding. And if I can't, that actually proves that you're correct.
Yeah. So when you're replicating somebody else's study, so
Hmm.
you're on the right track logically, but to kind of like work through it. But when you're replicating somebody's study, you use their original hypothesis and
Oh,
conduct
okay.
your own study.
Okay.
And then usually when you're replicating somebody's study, it's not to try to catch them doing something wrong. It's like if what Sean just found is true, then that opens up these other avenues that would be really interesting to pursue. So I want to pursue this other avenue, but I'm going to replicate Sean's study first to make sure I can find it in my lab. And then if I find it, then I can
Hmm.
do parts B, C, D, and E. But I can't do that without replicating your study first. So then they try to replicate you and go, wait, we didn't get those results. And a lot of times they'll do it again and again, maybe 3 times, and then go, okay, something's wrong. So that gets published to say we tried to replicate this and couldn't. And then usually you talk to the other research group, whoever it is, and say, hey, we couldn't find
Hmm.
these results. Can you look at what we're doing? Tell us how we're doing this differently than you did. So I talk about getting caught and embarrassed, but it's not because everybody's out trying to catch me. It's just people are going, oh, that's awesome. Then I'm going to build on that. And then there's no foundation there to build on. They go,
Right.
wait a minute. And that's when you get caught, if that makes sense.
It's yeah, that makes sense. Again, logically in my brain, I know it kind of sounds negative, but I don't mean it in a negative way. Even if I'm doing the initial research, not even the replicating of another study, but in my brain, the way that I make it make sense is that I have a hypothesis and I'm going to try to disprove it.
Yes,
And
that's
if
right.
I can't disprove it, that means that my hypothesis is not a hypothesis. It's a fact. But let's go back to then to results, right? So
Okay.
there are you're conducting this research. So I don't mean to drag us down too far, but go back. I have a hypothesis. I want to test it. I'm going to figure out how I'm going to set up the research.
Mm-hmm.
So you've already talked about like what kind of research I'm going to do, how many people do I need, and then those sneaky factors of time of day, of you call them variables, I think,
Mm-hmm.
but these variables that if you don't really obsess about the variables, it feels like you'll do the research and at the end be like, oh no, we missed it, we didn't even think about this thing, and so trying to get ahead of all the variables and cut those out, and then you conduct the research, and at the end of the research, um, whatever the findings may be, there are different kinds of findings, right? Meaning, like significant versus can you kind of you
Non-significant.
did the research.
Yeah.
Now what are you looking at?
You've collected a set of data, whatever they are, whatever those numbers are, and now you've
Hmm.
got to, so you can see that they're different because it's just, I used to tell students in my stats class when I was teaching statistics, if I divided this room in half right now and gave you all a pop quiz, and then the left side I average your score and the right side I average your score, they wouldn't be exactly the same because everybody in this room is different. So some of you just understand this a little bit better than others. Some of you study more and read more than others. There's some of you might have missed breakfast this morning and others didn't, so you're not thinking quite as well. So there's a lot of reasons of why you won't score exactly the same. So researchers expect there to be differences between the control group and the experimental group. There's always differences. So then we have to find out maybe I take the left side of the classroom and I have them read uh, uh, War and Peace for an hour. And then I take the right side of the classroom and have them study some notes I've created for statistics for an hour. And then I give them the quiz. Now I expect my hypothesis would be that the right side of the class is going to do better on the exam because my treatment, which was studying statistics instead of reading War and Peace, my treatment should make a difference. So now when I test them, my hypothesis would be that the null is that there's no bigger difference than there was the first time I just did a baseline where I just tested both of them, the right against the left. But now I can look. So now I need to know. Now I've got their scores back from the exam, and they're different because I knew they would be different. But are they different enough that I can say my treatment was effective? And so that's where you start into data analysis, and you try to find out, how different do these scores need to be before I can say that what I did had an effect. And there's been luckily there's a set out there, tables full of scores of differences that you need to find in order to say this was a significant difference. And significant can mean different things. So like in behavioral research, if I can be 95% sure that my treatment effect is what made the difference, then I can say that's significant. If I'm only 90% sure, I still have to say that could have just been due to chance. Maybe this, one kid on the right side of the room is just so much smarter than everybody else that's why their scores are better. And so, and there's ways to then pull outliers. There's so many different tools that you can use in statistics. But the goal is to find out, like in social and behavioral type research, to be 95% sure that your results are not just due to chance. And so that's the level of significance we set. In medicine, a lot of times, especially in drug studies, you have to be 99% sure that it's due to the effect of the drug and not just by chance. And so the level of significance changes, called the alpha level. So alpha 0.05 means that's 5%. So there's only a 5% chance this was due to chance at alpha 0.05. At alpha 0.01, where medicine is, there's only a 1% chance this is due to chance. And so you set your significance level before you start. And then when you come up with your findings, you report what your alpha level was, what your confidence levels were. You report if it was significant or not based on those. And so you'll read research where it's, 0.05, and you'll read some research where it's 0.01. And then you can also report, which most people do, the p value, which p is probability. So what's the probability this was due to chance? So instead of just setting a bar and say I crossed the bar, just what was the probability? And so that might be 0.0001. And then you're going, wow, I'm really confident that that's not due to chance, that finding. So that would be significant. If you don't reach that, that doesn't mean that your treatment doesn't have an effect. It means your study didn't find it. And so you don't, sometimes I would catch students doing this. They would say, my results are insignificant. And I would say, no, that's a value judgment, whether it's insignificant or not. They're not significant or they're non-significant. It's not, so you're measuring it against the bar you've set. Did you reach the bar? If you passed it, it's significant. If you didn't pass it, then it's non-significant. But it's not insignificant because what you did was meaningful. So, but if I say
words matter,
yes, and if I say, well,
words matter,
my test only came up showing that I can only be 92% positive that this wasn't due to chance, but I set my bar at 95%. I have to say my results are non-significant, but there was a trend to show that this does have an effect, which means I can go research that again because it might have been an outlier, or other people can. So that's still publishable. You can still publish that because other people will say, okay, well, I see what they're doing, but maybe if I change this or that or tweak this a little bit, maybe I can reach significance. And so you can do things like if I'm testing a drug and I give somebody or I give a rat, let's say, a placebo to one group and 5 milligrams of this drug to an experimental group, and it comes out to be like 97%, that you're 97% confident it's not due to chance, but that doesn't reach the 99% bar, then maybe you redo the study and do a placebo group and a group with 10 milligrams of that drug, because then maybe that will reach. And so that's why a lot of times with drug studies, you'll have multiple experimental groups with 5 milligrams, 10 milligrams, 15, and look at it that way.
Yeah. And so now I'm thinking about it's a long running joke, but the ads that I see on TV for drugs and the side effects that go along with that drug.
Yeah, it just
that's more of a liability thing, but
yeah, but it is, I think, a liability thing, but that also, you have to be careful, because that doesn't mean that this drug causes everything you're hearing, which is what a lot of people think. It means that during this trial run, somebody ended up with diarrhea, somebody ended up with bleeding, somebody ended up, but it might not be because of that. It's just, like I said, everybody's different. There's different reasons, and drugs affect people differently. The same drug affects each one of us differently.
So it's interesting you just said that, because that's a little bit about the direction I was going with that, is that the list of side effects, negative, it could be viewed as negative side effects, doesn't mean that everybody in the trial got those things. It doesn't mean that one person got all those things. You just said it, but I really want to bring it back to, like, we did this trial of this drug to try to treat whatever thing, and during it, this one person got diarrhea. This person over here had a headache. This person had, whatever it is.
Person couldn't sleep.
And then so you went, and the horrible one, the suicidal thoughts, or there's some other ones I've seen in the disclaimer, but it's not everyone in the trial got those side effects.
No, and then
even though that commercial, it makes it seem like if you take this drug, you're going to get all this.
Yeah, exactly. And I think even important, like you said, not one person got all of those. So not only did not everybody get those, but yeah. So if you run 2,000 people through this study and 3 people get diarrhea, you have to list that as a potential side effect. And 12 people got headaches, but what you didn't know is that when they reported a headache, they hadn't had their coffee that morning or something. But you can't go back and try to control that. You just say, oh, you had a headache? Okay, we have to list that as. But that's not to say that these side effects aren't real, because sometimes they do cause these things. It's just when you first hear it, you can't assume that that's going to happen to you or anybody in particular.
Yeah. And like I said, there's a lot of comedians and a lot of skits about the side effects are worse than the thing that, I'm taking this pill for, or whatever the treatment is. But again, it's just important to point out, like, research. Why I really wanted to talk about this with you is I think that, I don't understand much of this, and I've been involved in some research with you. When you talk about the p-value of something, oh, okay, so the farther out the number is from the decimal point, the less likely that this has to do with chance, right? I mean, and then again, you're running these different kind of trials or this different kind of research, the difference between behavioral research, medical research, what are some others? I guess those are kind of the big ones,
they usually relate to one of those two, like physical changes to you, or emotional changes, or cognitive changes, kind of are the things you're looking at in experiments.
Okay. But the research, especially today, is there's so much thought that goes into it. You already mentioned this too. I was thinking about your friend that hopefully I'd say our friend, but who is going through research right now. And because they have this rare form of an aggressive cancer, um, that it's probably not as researched as the more common variations because of funding. Even before what's going on right now with funding and science and the like is like you have to, or maybe you don't have to, maybe you can answer this, but like you have to devote the time and energy to the more common cancers to try to deal with that. Just from a statistical standpoint. I mean, not to be callous, but like not as many people are suffering from this. So let's throw all of our resources at this other thing. And so now we've decided, okay, we're going to research this particular thing. Hopefully we have the funding for that. And then we don't want to, I don't want to say it, but we don't want to mess this up. Like, let's really be thoughtful about this and try to do all this. And then when, again, setting the parameters for the results, and then we look at the results and, where you just said it, we set the bar at 95 and we hit 92. And again, I like that you said this, it's not a failure. There was something in there. Whether we didn't set the bar the right way or there was some another anomaly or whatever it is. And then either I can redo that research or we publish it and say we hit 90, we didn't hit the mark, but we did this. And then someone else might be able to try to replicate that and fix it. I mean, I know it's kind of a rambling statement, but like, that's what I'm interested in. That's what I wanted to learn about.
Yeah, and
all of that.
Even if you say this reached significance because you passed now you're 90, like if it's in social-based, behavioral-based research where you set the bar, you want to be 95% sure. That's where I'm going to set my bar. Even if I'm 97% sure, there's still a 3% chance that was just due to chance. So it still could be that it can't be replicated because it was just due to chance. So you can never be absolute sure in that sense, but that's why you have to set it somewhere. And that's why it's set so high so that so there's not as many, false alarms that we just found something that's not there. So there's more, like you might miss something where if it's 92% and you just say, I'm going to go a different route, there might have been something there. You missed it because it didn't reach the bar that you set. But yeah, those are chances in research always.
Yeah, because I'll tell you, I was listening to Revisionist History, the podcast by shout out Malcolm Gladwell. I love his podcast,
great.
But and they weren't talking about research, but what they were talking about is raccoons versus rats.
Oh yeah,
because and I don't want to recap the whole episode on here, but what struck me is that raccoons, because they have such a hellacious raccoon problem in Canada, in Toronto specifically, but raccoons seem like they can solve problems really well. Like they can get into stuff, and they made these raccoon-proof boxes and spent millions of dollars to the Toronto local government to set them out. And the raccoons, it took them like half a day to figure it out, and they can get in there because there's food in there.
Yeah.
And so the idea is like, why are we experimenting on lab rats, when raccoons seem so much more intelligent? And then, and again, in the episode, they did a good job of breaking this down, but it's like raccoons aren't actually that intelligent. They are just they're good. They're going to get in there, right?
They're
so they'll figure it out to get the food. Yeah. They seek and destroy.
Yeah.
Right. And so it's like, and then what you just said to me today about rats, I have these thoughts, John. I think I have things figured out, and then, and then I bring them on here, and then just, yet again,
but that's
Sean,
that's common. Even people in science who are or outside of science, even academics who aren't in science have the same questions. Like, why do you all I'll be at a party, a get together. I'm not now, but when I was in academia, there'd be like a get together, and you'd be visiting with people from music and from history and whatever, and they would say, like, why do you guys
Hmm.
research with rats like that? Those aren't humans. Why do you do that? So it's a great question, but there are
Okay,
answers.
so
There
I'm
are
not the
answers.
only one.
No. And
I'm not the outlier.
no, you're not at all. But I think maybe I could even drill down a little bit more on that. So
Okay.
so there's genetic similarities between mammals. Like chimps were 98 to 99% similar. Other apes are 95 or more. But with mice and rats, we're anywhere from 85 to 90% similar to mice and rats genetically.
Really?
And
Okay.
like
Okay.
dogs, we're 85% similar to dogs. Again, because we're all living in the same sphere, the same world that share some of the same
Hmm.
things. And what's also interesting is the structure and function of those genes are largely conserved, meaning that not only are we similar genetically, but the genes tend to do similar things. They're expressed in the same similar way, which makes it easier. And then neurotransmitters are remarkably similar between mammals. And again, it's because I mentioned like our diet. Food is the precursor for
Hmm.
neurotransmitters that our brain uses, well, that our body uses too, but we're talking specifically about the brain. So food,
Yeah.
what we eat is kind of the precursor that gets converted into the neurotransmitters that our brain uses for food, for fuel. So broadly, neurotransmitters are like amino acids, soluble gases, neuropeptides, but some of the ones that people are more familiar with, like dopamine, serotonin, acetylcholine, norepinephrine. Those, if we're looking just at rats, dopamine, which is used for like motivation, reward, learning, those types of things, and movements, are nearly identical in distribution and function in the basal ganglia and the prefrontal cortex of rats and humans. It's almost a
Hmm.
mirror system. Um, uh, Glutamate, which is the primary excitatory neurotransmitter, has identical roles in like learning, long-term potentiation, learning between rats and humans. GABA, which is a primary inhibitory transmitter, it has it's very similar in its circuitry and how it's used in its circuitry. So those are the neurotransmitters. When we're looking at drugs, that's where drugs have their effect. So neurotransmitters leave one cell, like one cell will spew out some neurotransmitters into the synaptic cleft, what am I trying to say? The liquid in the brain between cells. It'll
Hmm.
spew these neurotransmitters out into that liquid area, and other cells will take that in. And so then that's how messages are converted.
Messages. Yep.
But
Yep.
drugs, there's also enzymes in that synaptic cleft that will start like chomping down and eating the neurotransmitters before they can get to the other side. There's neurotransmitters will get spewed out, but some of them will come back and be taken back in by that same cell, so that's reuptake. So drugs will have their effect on like the enzymes that break it down, or they'll block reuptake, or they'll block uptake, or there's just different things that happen. And that's similar in rats, mice, humans, monkeys. It's
Wow.
almost identical. So that's why you can then test out drugs and things in rat and mouse models, and then say this is probably how it's going to work in humans. It also, I mentioned it allows for tightly controlled experiments. So that's the
Hmm.
genetics. So now I can create a mouse model of autism. So there's mouse models of autism. So mice that are pre-genetically determined to have these characteristics, they're modified in a way that they have reduced social interactions. They kind of stay to themselves. They have increased repetitive behaviors. They have altered synaptic development. So all of that's really similar to humans with autism. So now researchers can use these mice and look for and study the the genetics behind it, the neurobiology of autism, and then if there's an idea for a potential treatment, it can be tested in the mouse models first. And but there's it's other things besides just that. I mean, there's like some of my favorite studies I would tell students are like the rat park study. This was in the 70s and 80s. They were looking at the effect of drugs and addiction, and so they would put morphine in water, so it'd be like a drug laced water, and they would house some rats. They would isolate them by themselves in just a cage with really no toys to play with, where they're just sitting in a cage by themselves with this morphine laced water. And then they would house other rats together in a community with a lot of a really enriched environment. So there's little balls to roll around, there's things to chew on and play with. And then they would see how much morphine each rat consumed. And it's not surprising that those that were living by themselves consumed significantly more morphine. In fact, rats in the rat park, they called it a rat park because they were living in a park-like setting. Some of them ignored because they had regular water too. So they had regular water and morphine-laced water. They would just drink regular water and skip the morphine. But those that were isolated almost always just started drinking just the morphine-laced water. So then it started telling us about addictions and how important, social connection is and environment is and having a purpose. So it's more than just drug studies. There's experimental studies like that where we can see real similarities.
So I don't know if it's dopamine or not. I don't know what it is. But if my phone makes a noise, that I have a notification, mm-hmm. and it doesn't matter what the notification is, it just ding. I have to look, and when I see a notification from anywhere, I have to clear that notification. I have to.
Yeah.
And then once I clear it, no matter what it is, I clear it. I feel okay.
It's like,
okay, now I can.
Like relief.
Yes.
A flood of relief. I'm the same way. Yeah. I think that's why apps have notifications to motivate you to do something about the ding, or even if I don't hear it or feel it ding, if I just glance down and I see up at the top of my phone, there's that little circle from that app that has it's like, I got to see what it is.
I need that to go away.
Yeah.
I need that to go away. So I'm saying that only to say I started this episode wanting to talk about rats and humans. And I'm just a rat in a cage. My cage is just bigger. I'm pushing the button every time I get a thing that tells me to. So
yeah,
you've convinced me.
Well, and then I think we should also probably touch on the ethics behind it a little bit. So because there's ethical and practical reasons to use rats and mice. So like some invasive techniques, like implanting, electrodes in the brain, is not ethical in humans, and it's done in humans if somebody is already scheduled for brain surgery. So like if somebody's having seizures and this is rare too, but sometimes they'll perform surgery and implant an electrode in the brain to try to control the seizures. So while the electrode is there, they'll ask the person, while it's there, can we also run some of these other experimental tests? And, if the person says yes, then they can use those electrodes to measure some other things. But that's really...
But they were already going to be in there.
They were already going to be there. And that's really, really rare.
Yeah.
But with rats, with monkeys, you can use a more invasive technique, to find out some of these things in research. But I also want to mention on the ethical side that there are really strict guidelines on what researchers can do. So and I'm just speaking from, most research takes place in higher education institutions like universities, colleges. There's also pharmaceutical companies that do research, and there's a little bit of research outside that as well. But I would say the bulk of it is in higher ed institutes, and that's where I came from. That's where I was doing my research. So I'll kind of talk about that. There are 2 committees in every university that conducts research. One is called an IRB, which is an internal review board, and it's made up of it's made up of faculty members, sometimes administration, sometimes a student representative will be on there as well. In fact, I think most of the time. And it has Washington DC and one of the federal agencies responsible for overseeing research. I should know the name. It's slipped my mind right now. But they set out like general guidelines for universities. You have to meet these guidelines. And so this IRB, this internal review board, makes sure those guidelines are met. And this is how you treat human participants. So the IRB is for humans, for college students. So, psychology departments use this more than any other department because they running tests. And that's why I don't know, in your intro to psych class, did you have participation in research a requirement or for extra credit or something like that? Because most places
it was extra credit.
Okay, most places either require or have extra credit because there's so many studies going on. We need participants, so we'll give extra credit for that. Then the other committee is called the IACUC, which the acronym is IACUC, and that stands for Institutional Animal Care and Use Committee. So IACUC, Institutional Animal Care and Use Committee. And that's also made up of faculty and maybe administration. And I think there's usually a student representative, and there's usually a representative or two from the community, so outside of the university. So I served on the IACUC board when I was at Dickinson College. And it's usually biology departments, neuroscience departments, and psychology departments use IACUC. Sometimes chemistry, but it's usually those groups. But there are really strict guidelines over the use of animals because, with the IRB, one of the main requirements is that you get consent, informed consent. You tell a student what the study is about and ask them if they agree to participate. So you have to be upfront and tell them what's going to happen to them. They usually read a paper and then sign it, I agree, but you can't get that from a rat or a monkey. So humans have to oversee that. And there's a really lengthy course you have to take, an exam you have to take and pass it to be a member of this committee. And you have to, renew it annually. And it makes sure that you are not doing animal research frivolously, and you're not putting animals in positions where they are, in pain or things like that. And if it does involve some type of invasive surgery or some type of pain, then there's a fairly lengthy process you go through to find all the studies that have done something similar to you that use these invasive techniques or cause pain, and then describe those and tell why your study is different and why you should have permission to continue this instead of using the data that's already there. Because there's no need just to continue to do the same thing over and over to rats. There has to be a good reason why you're wanting to do something similar. So I just wanted to mention that it's highly controlled, and there's great care given to make sure people aren't just doing surgeries on rat, all of that.
Right.
If that makes sense. I just thought it'd be important to mention that.
Makes sense. I think that's a good capper to end with that. The ethics involved are taken just as seriously as the structure, the results, all of that.
Yes, it is.
Yeah.
And if you do research, any type of grant funding has to be IRB or IACUC. Any type of publications, the journals require you to what would you say, to have followed those. You have to state that I followed IRB or IACUC protocols. And so, it's really important.
Wow. Oh, John, thank you. So listen, I'll be honest with you and the listener that we have left. Of all of the episodes that we have done, this one was for Sean. I wanted to learn more about research, and this one was for me. So I appreciate you taking the time to explain all that.
Yeah, and you've been involved in research before, but you kind of came in at the other way, where instead of going to school and having to learn to conduct it, you joined a research team, which is a great way. That's actually I think you probably learn about it. It might be a better way to learn about it is to just jump in and join in some research, but then you probably missed some of the things we talked about the design, some of the
I
other things
did.
behind it,
Yeah.
which are also, that's not hard to catch up on those things, but yeah, it's all a part of it. So
Yeah. It makes so much more sense to me right now. I'm very
good,
fulfilled. I
good,
appreciate that.
good deal.
Uh, and yeah, and for everybody else, don't forget our socials, TikTok, Blue Sky. John's TikTok is still. I keep wondering if you're going to run out of steam, John, but you don't seem to be slowing down. Your content on brain_dude, right?
Yes. Yeah.
But that I just crack up. Those TikToks are keeping me content.
Good.
So
Yeah. I've had to get a few extra neurons firing to come up with some of that, but yeah.
well, that's good. That's good too, though.
Yeah.
And yeah, so everybody else, thank you for listening, and wherever you are listening, hit that like, subscribe, favorite, whatever it is, wherever you're listening, and don't forget to tell a friend or two, a coworker, a family member, anybody. And until next week, take care of yourself, and we'll see you then.
Brain juice Sean.